Enjoy this Aztlander presentation, Insights into the History of the Americas, as revealed by ancient DNA with Nathan Nakatsuka.
So first, I'll just start with some background on genetics and ancient DNA. And so just a high overview of genetics. It's just a reminder about basic genetics. So all humans have 23 chromosomal pairs, with 23 pairs from your biological mother and 23 from your father. So you pass down one pair of these chromosomes through each generation, but each time it is passed down, there are random mutations and recombinations that lead to changes over time. So this process makes it such that individuals who are more closely related to each other will have fewer genetic differences between each other. And the same goes for larger populations, where groups that are more closely related to each other will have more similar genetic sequences. Since these patterns of DNA are being passed down in very predictable manners, you can examine the DNA of individuals to see how they relate to each other. And this is, for example, what you do in forensic cases to determine who is at crime scenes. And you can also look at the DNA of larger populations to see how they relate to each other or what population specific individuals are from. So ancient DNA is DNA from humans or other organisms that lived many years ago, often thousands of years ago. So this is obtained from skeletal material of the ancient humans. It's brought into a clean room to minimize contamination from modern humans. And a small amount of the bone is then powdered, and DNA is purified from this material and then sequenced. So what's the big advantage of ancient DNA compared to DNA of present-day humans? The big advantage is that now we can observe genetic variation not only in geographic space, but over time. So you can compare the genetic profiles of individuals from the same region over time, and you can tell how the genetics of the area is being changed over time, such as new people coming into a region. So one thing a lot of people study is the mitochondrial and Y chromosome DNA. Mitochondrial DNA is DNA that's passed down exclusively from the mother. So it shows human history from the maternal line. And in contrast, Y chromosomes are only found in males because the sex chromosomes of males are XY, whereas females are XX. Analyses of Y chromosomes show human history from the paternal line. So here's a figure of this. In the case of nuclear DNA, it's inherited from all of your parents and grandparents, but mitochondrial DNA is only inherited for the maternal lineage. So you're all the females going down through history. So in contrast to mitochondrial DNA, Y chromosomes are inherited only from the paternal line. So mitochondrial and Y chromosome DNA can reveal some aspects of human history, but the autosomes, and these are the non-sex chromosomes, are usually more powerful for revealing mixture between two populations because they're much larger and there are more of them. So I often use this example when teaching Native American high school students, some of whom might be, for example, part Navajo and part Hopi, or part Cherokee, part Shoshone, or etc. And so if you have your child, say from a Navajo mother and a Hopi father, then you'll have one set of Navajo chromosomes and another set of Hopi chromosomes. And each successive generation, their chromosomes become more mixed together into smaller and smaller pieces. And you can use various statistical analyses to infer these events and also when they happened. So there's a very important quality control steps for this process, but one of the most important ones is reducing contamination. So you need to ensure that the DNA is human and not bacterial, and also that it is ancient and not from modern human contamination. To ensure it is human, we map the DNA sequences to the human genome. And to ensure that the DNA is ancient, we look for DNA with characteristic damage. There are multiple other ways to determine if there is human contamination. For example, we can look at alternative ancestry, such as, say, European ancestry in ancient Native Americans. Or for male individuals, we can look at heterozygosity, which are two different types of variants on the X chromosome, since males should only have one X chromosome and therefore no heterozygosity. We also developed this tool called ContamLG, which works for both males and females, and it's based on the breakdown of what we call linkage disequilibrium on the autosomes. It's a bit more of a complicated method, but it's a more powerful method than the just the X chromosome method. So to make historical inferences with ancient DNA, you usually need data of very sufficient depth and quality. This usually means over 200,000 SNPs or single nucleotide polymorphisms. These are genetic variants. And it's helpful to have many individuals, but even one individual can tell you something, such as whether a particular ancestry type was present. More individuals helps to give you more statistical power for analyses. And eventually, if there are enough individuals, you can start seeing genetic heterogeneity within a site. For example, I live in New York right now, and say there's an alien who comes in maybe 20,000 years from now, looking at the DNA of people living in New York City now. They might get a single ancient DNA of someone in New York, and they could say, maybe there's someone who's of Italian ancestry there. And then they start getting more and more DNA samples from New York, and they'll see there's Peruvian ancestry, there's Ecuadorian people here, there's Chinese people, et cetera, et cetera. So the same thing goes for if you're doing it in an ancient Native American site. There might be people from all over the place in a particular city or not. So you can see that based when you have lots of different individuals. So I'll first start with our study on the earliest peopling of Central and South America. This was an enormous effort with many institutions across the world involved, but it was especially driven by many of these key collaborators: Cosimo, Elsa, Lars, Johannes, Gustavo, Andre Strauss, Doug Kennett, and Cesar Mendez. So before I begin about this study, I'll provide some background on some of the past genetic studies of ancient and present-day Native Americans. So the formation of Native American ancestry actually goes before the crossing of the Bering Strait. The Native American ancestry first began forming around at least 20,000 years ago, when an East Asian group mixed with the North Eurasian group in Siberia. And so there was mixing, and then people were basically present in Siberia for long periods of time. And the ancestral stream giving rise to most of the Native American ancestry today is a mix of about 2/3 East Asian ancestry and about 1/3 of this ancestral North Eurasian ancestry related to this individual in Northeast Siberia. So individuals with this ancestry came into the Americas about 16,000 years ago, maybe even more. And there was an early split of the ancestry, with ancestry A being found in all Central and South Americans, as well as many ancient North Americans. We just call it ancestry A just because it's just a name that we're giving it. And there's another ancestry type, we call it ancestry B, that's found only in Canada, Alaska, and Maine, and is present in groups like the Algonquin and Athabascan speakers. And in the north is some additional East Asian ancestry in some of the northern groups like the Inuit and Aleutian Islanders, which is due to additional gene flow that came from East Asia and from Siberia. So the genetic structure of present-day Native Americans generally correlates with geography and language group. So here you can see a genetic tree of modern Native Americans, and here are Eskimo-Aleut and Inuit groups in Greenland, and then you can see Athabascan and Algonquin groups in Canada, and Central Americans in Mexico, and further south you can see people from Costa Rica and Panama. And you can see the rest of the structure in South America with Peru, Argentina, Brazil, Colombia, and other areas all separating. So this study focuses, that I'm going to talk to you about, focuses on the genetic structure and history of the oldest Central and South Americans. One very important part of ancient DNA projects is to work very closely with the living communities associated with the ancient individuals we are analyzing. So unlike studies of present-day individuals, you're not able to get consent of the individual you're studying, but similar to studies of recently deceased individuals, in, say, medical context, you might get consent from the families of the individuals. And in ancient studies, it's similar, where you're engaging with the local communities, because the process of the study and the findings can sometimes have the greatest impact on them. So throughout this study, we consulted with indigenous groups near each of the areas where the archaeological sites were. And this varied in different areas. So in Peru, much of the country views themselves as directly connected to the indigenous history. So we consulted first on a governmental level and with local community archaeologists, and then also with local communities that feel more attached to their indigenous ancestry, and with several public presentations of collaborators who were able to speak in Spanish and Quechua and Aymara, and those were given in Lima, Cusco, Bolivia, and other areas in Peru. So there's also museum visits for the public to share these results as well. So in Chile, Argentina, and Brazil, we got research permits from the institutions near the sites and consulted with indigenous communities located nearby, such as the Comunidad Indígena Mapuche Tujote, living near the site of Laguna Chica in Argentina. And in Belize, we directly work with the Mayan communities, and our collaborators gave several presentations to the community members and got their feedback and permission for the study. So one of the first things that we observed genetically, which was very interesting, was a very rapid radiation of the earliest groups into South America. So basically people are, you can see this by the genetic differences between different groups being split off very quickly relative to, say, an area where there's farming, where people are slower to split. You don't see the same patterns there. So in this case, we see that people are really quickly, the earliest hunter-gatherers are moving really quickly to these different areas, and probably they're just chasing food and quickly switching to different areas to get there to follow food going downward. After the early radiation, there's much more continuity observed, though not in all ancient groups. So here's a map that shows the genetic affinity of the modern groups to this ancient Peruvian who is 8,600 years old. The ancient Peruvians in green, and the red indicates that there's more shared genetic drift. Basically, it means that there's more genetic relatedness of the people there, very close, living close to that area where that ancient Peruvian lived. And so this indicates that basically the people who are living in the area are very genetically related to the individual who lived there up to 9,000 years ago. In Brazil, you can see the same thing. In Argentina, Chile, you see the same thing. And in Brazil, you see the same thing where a lot of the people living near the area are more genetically similar to them. But it's not always the case. So there's some places where there's more spread out ancestry, such as in this Brazilian individual on the left, the Brazil La Palo Santo, it's more of a spread ancestry throughout the area, meaning that basically all the South American groups are about the same genetic ancestry as that individual. So Belize also, you see the same thing happen over time, where the earliest individuals don't have the genetic drift, and then individuals who are younger have the genetic drift or shared genetic ancestry. So basically what's happening is people are starting to stay in the area once you develop agriculture and other sorts of things that allow them to plant in the area, and the population grows and people settle down in that area. So you can also do the genetic clustering analyses of the individuals, and you find that the genetic structure of ancient Central and South Americans generally correlates with geography, except with some interesting exceptions. So here's, for example, a plot, a multidimensional scaling plot, where the individuals from the same regions are generally clustering together. So you can see Peru in green, and Chile and Argentina and Brazil in separate areas, and most of the clustering is geographic and not temporal. There's another sort of way you can plot it with the neighbor joining tree, and shows that most of the people from the same area are clustering together. The exception to this are some individuals who are clustering with this individual, Anzac 1 individual that was associated with the Clovis culture that Jim was talking about. And this individual is from Montana, and yet you see that there's a genetic clustering of people who are not at all close to Montana with that individual. So I'll explain that soon. So one way to do this is you use various statistical tests to test for heterogeneity of the ancestry of the South Americans relative to North Americans. And using that, we found that at least 3 different genetic exchanges occurred between North and South America. So one of the key findings is that ancestry related to this Anzac 1 Clovis culture individual started arriving very early on in South America. So it's not like necessarily that the Clovis culture itself went down there, but ancestry related to the Clovis culture is coming down into South America in the earliest individuals up to 11,000 years old and to 9,000 years old. And so it shows that ancestry related to this culture is spreading into Central and South America. And this ancestry is then mostly replaced throughout South America, which is really interesting, and suggests that there's an additional wave of ancestry that's kind of replacing a lot of this ancestry. We searched for additional genetic exchanges between North and South America, and there's another genetic exchange in the Central Andes where individuals in Peru have this extra ancestry after 4,000 years ago that is related to people with kind of Northwest Mexican ancestry that we found. So right now it's said as California Channel Islands, but actually it's more of a Mexican ancestry that's coming into the Central Andes. So after studying the oldest Central and South Americans, we studied 65 additional Central Andeans from 4,500 to 450 years old to determine the genetic history of the Andes, including the genetic changes that accompanied the rise and fall of the major cultures there, such as the Moche, Wari, Tiwanaku, and Inca cultures. The study involved many collaborators and institutions throughout the world, and it was driven by the local archaeologists. And some of the key researchers were Lars, Jose, Chiara, and Bastian. So when people first think of the history of the Andes, they often think of the Inca Empire and Machu Picchu. But what is less often realized is that in the Andes, there have been many other cultures and complex societies that preceded the Inca, including, for example, the Tiwanaku in the Titicaca Basin region, the Wari in central and south Peru, the Moche in northern Peru, as well as many others. And so in this study, we're tracking the genetic changes that occurred over time in the Andes, starting from some of the earliest hunter-gatherers to individuals who lived immediately before, during, and after the existence of the major Andean cultures. So we analyzed whole genome sequence data of 89 ancient individuals from 9,000 to 450 years old. So we first look at the oldest individuals from the North and South Highlands of Peru, and we found that these individuals were more genetically related to later highlands individuals in the regions relative to the later coast individuals. So it implies that there's a distinctive Andean highlands ancestry that was already in place by 9,000 years ago. So if you examine the northern and southern groups, there's no difference between them, but there's a difference between the highlands and the coast. After this, the north and south start distinguishing themselves after around 5,800 years ago. So at this time, basically, there's plant cultivation starting, there's increased sedentism and increased population growth, which could have contributed to this population structure where the north and south are now differentiating from each other. After 5,800 years ago, we observed migration between the highlands and the coast. So people are going up and down the highlands and the coast. And also between the north and south highlands, people are moving up and down the Andes. We find that groups in the Titicaca Basin have gene flow from the South Peru highlands and North Chile. And we also found that the Andes had significant genetic interaction with external regions. So approximately 2,000 years ago, Northwest Amazon ancestry spread to northern Peru, and particularly the northern and central coast. And in addition, the South Central Andes had genetic interaction with Argentina, even impacting the Pampas region over 2,000 kilometers away. However, after 2,000 years ago, we find surprising genetic continuity in most of the regions, despite constant cultural change and the succession of major cultures. So the Moche, Nazca, and Wari are very large cultures that you would think when they fall down that there's going to be a large genetic change accompanying it. So for example, in Europe, lots of times when these cultures fall down, that usually it means a large influx of people are coming in to either for war or other things. But this is suggesting that in Peru, the transitions of societies and archaeological cultures was not accompanied by massive population movements in these regions, and might have been, say, due to climate change or other sorts of reasons. We did find exceptions to this continuity. For example, we find that individuals that lived in the capital of the Tiwanaku polity that dominated the Titicaca Basin around 1,000 years ago had more ancestry related to southern Peruvian highlands relative to individuals from other Tiwanaku sites in the Titicaca Basin. And we even see Amazonian ancestry in the capital, even though the Titicaca Basin was more genetically homogeneous. So around the lake, it is more homogeneous, but at the capital, where they're doing human sacrifices and other rituals, there's a lot more genetic diversity. So this could be due to a lot of different reasons. It could be due to their taking in people might be coming in for work or other reasons. And I'll show you also that the same thing happens with the Inca Empire. So during the Inca Empire, we find cases of individual migrations of very, very long distance. We observed that similar to the Tiwanaku capital, in Cusco, which is the heartland of the Inca Empire, this harbored people with diverse genetic ancestries. We find individuals from the Peruvian North Coast and Titicaca Basin all coming into Cusco. And so it seems the capitals of these societies are more cosmopolitan than the rural regions, with people with different ancestries who are living side by side. This might be similar to, say, New York City or Boston or other places where there's a lot of different ancestries in the cities and maybe less diversity of ancestries in other areas. So some of these migrations are not necessarily by choice. So we know from historical records that the Inca displaced and relocated individuals and whole groups from their original territories. And we find a clear example of this in the genetic ancestry of a young child who was sacrificed during the Inca period on an Argentinian mountain summit. the boy had northern Peruvian coast ancestry, meaning that he came from the north coast, which is thousands of kilometers away from the place he died. And the same north coast ancestry you find not only in Cusco in Argentina, but also in the southern Peruvian coast at Chincha Valley. And the genetics here is consistent also with Strontian isotope data and north coast style textiles and pottery that is found in the area, as well as historical written records of Inca moving people to the area. So for this project, I'd like to acknowledge the Peruvian, Bolivian, and Chilean communities we worked with during the study, as well as the Comunidad Indígena Mapute Tuolche and the Mayan communities and the Yakshe Conservation Trust in Belize and the National Institute of Historical and Artistic Heritage in Brazil. There's many others who contributed to this work, especially those I mentioned earlier, and many others, including Doug Kennett, Brendan Culliton, and Thomas Harper, and others. So this last South America project I'll talk about is about Patagonia, and this project was driven by the indigenous Patagonian groups, as well as Rodrigo Norres, Pierre, Monica, Gabriela, Josefina, Ricardo, and Carlos La Luisa Fox. So this project was really interesting. We're able to investigate the genetic structure of Patagonia over time. We find that the development of the marine-based economy, so basically in Patagonia on the western coast, there were people who basically hunted sea mammals. They would throw harpoons and other things to get these sea mammals. They're also, in general, much shorter than the people on the east side who ate more land animals. They were a terrestrial diet. So there's a very big difference between these two people. And we wanted to, two people groups, basically the West and the East, and we wanted to see whether these canoes and the harpoons were due to genetic change, or they were they made in situ. And we found that they were actually due to in situ development of these technologies, because we find that the individuals have very similar genetics at the very beginning, when the first individual that who is around 6,600 years ago, who had a maritime diet, and the first individual that we have who had a terrestrial diet have very similar genetics. Whereas genetic changes started happening after that, when people started migrating from the north, and this correlated with different archaeological changes, such as there's a certain time period when there's abandonment of green obsidian use around 4,000 years ago. And then there was an additional change in the transition from boleadoras, which are basically these ball things where you throw them around the legs of animals. And at a certain point, around 2,000 years ago, there was a switch to lithic points, and we see a genetic change at the same time that's happening at that time, suggesting that people are arriving in and bringing in these technologies, and people are coming from the north to bring in these technologies. We also see gene flow that's going around the coast, where basically there's a gradient from the west here all the way up to the east, and individuals, particularly the Hausch here, who use both maritime food methods as well as terrestrial methods, have a mixture of ancestries from the east, who only do terrestrial diet, and from the west, who do only maritime diet. So we had a lot of data from all these different groups, and we're able to see a lot of interesting things in Patagonia, which you could ask me more about as well. So I'd like to thank the many collaborators on this project, especially the ones I previously mentioned. So now I'll end with a California project. First, I'll start with some background about ancient California. So first, I'm going to start talking about why this region we now call California is a very unique and important place for Native American history. So the history of the people in the Americas, as I said before, began around 16,000 years ago. And it was thought that one of the first routes they traveled likely was along the western coast, with some of the first people migrating near the ocean. And another group might have come down several thousand years later through the middle of the continent, associated with the Clovis culture. And there has been evidence of a very early migration. For example, we have archaeological evidence of people living in the Santa Rosa Island from at least 13,000 years ago, based on human skeletal material, stone tools, and other human artifacts. California is really interesting because there's, as I said before, there's extremely early peopling of California, which is evidenced by the great diversity of the different languages in the region. So for example, in Europe, where almost all the people in Europe speak a language in the Indo-European language family, there are many In contrast, many highly divergent language families amongst the indigenous languages of California with split times over 10,000 years old. So for example, in Northern California are some of the Algonquin and Athabascan speakers. And in Central California, which is the region where San Francisco and Palo Alto are the Yuxian languages, such as Ohlone. And in addition to the Yuxian languages, a few linguists have also proposed an ancient language family called the Hokan language family, which it could include languages such as Aztlander. And there's another large language family in Southern California called Uto-Aztecan. Some examples of these languages are the Gabrielinos, who are also known as the Tongva, and they reside in the area we now call Los Angeles, as well as the Payonka-Wichem or Luiseno, who mostly live in San Diego. So here's a broader map of the Uto-Aztecan languages. There are multiple hypotheses for where the Uto-Aztecan migrations came from. One hypothesis is that it came from people came from central Mexico with farming, and then farming was then lost in California in the Great Basin. So some linguists proposed this. Peter Bellwood and the late Jane Hill, who is a linguist, were major proponents of this hypothesis. Another hypothesis is that the Uto-Aztecan languages originated from the Sonora Desert, from hunter-gatherers between 3,000 and 5,000 years ago. Lyle Campbell previously proposed this view, though he might have changed now to propose the Southern Arizona view. Others believe that the Uto-Aztecan languages came from the Central California Valley, which was proposed by David Schall. And William Merrill and several others proposed the Great Basin as origin. Some others have proposed, as I said before, Southern Arizona as a source population. So it's very confusing for where the Uto-Aztecan language is. And most of these arguments are linguistic arguments based on loan words and different cognate roots, and whether you can construct an agricultural vocabulary that encompasses all of the languages. So there's a lot of different debate here about where this language family came from and how much interaction there was between the different groups, and when they arose, when they split, etc. So ancient DNA is one piece of evidence that can be used to provide evidence for and against some population movements at different time periods, which can be relevant to the debates. California indigenous groups also have a wide array of oral history that informs views on how people were created and how humanity relates to nature. For example, the Chumash oral history talks of an ancient rainbow bridge that allowed people to move between the areas that was created by Mother Earth, or Hutash, which promoted cross-island travel and travel between the islands and the mainland. They also have an alternative creation story based on the Meshupashu, creating people from a marble table. Ohlone have an oral narrative about creation after a flood by either a coyote or a feather from an eagle. Tongva have a creation story about the first man and woman and guidance from the Quawar, who received the souls of all who die. And so basically, these oral histories usually show a strong connection of the people to their homelands, but also with resilience to change. So we discussed these topics with indigenous groups and archaeologists and arrived at several key questions we sought to address with this study, including were there interrelations between California and within California with California and other regions? And how did the people on the Channel Islands relate to those on the mainland? And how much genetic continuity was there over time? And when did the different language families arrive in the region? What were the original source populations of the language families? And additional future questions we could ask are, were there smaller population movements that happened in recent history, and as well as the population sizes, and how do the ancient people relate to living people today? I'll get to that question after, yeah. So previous to this study, the only whole genome studies previously published on ancient California were in 2018 and 2022. The 2018 paper focused on broader global Native American ancestry components mixing rather than more recent history, while the 2022 study found evidence for genetic continuity in Central California over the last 2,000 years. So in this study, we studied data generated from the prior studies, as well as 80 additional ancient individuals who lived in Southern and Central California, and 40 new ancient individuals from Mexico, primarily from the Northwest region. Prior to the study, we consulted with indigenous groups in the regions near where the ancient individuals were found, including several Chumash tribes and Tongva in Southern California, and Ohlone and Aztlander communities in Central California, as well as the Mexican government and indigenous Mexican communities in Sonora in Northwest Mexico. We also held several meetings to return results as they came in to ensure that the study was conducted and the results were reported in a way that centered Indigenous perspectives and the questions they were interested in. So here's a map of where the ancient individuals in the study lived, encompassing Central California, Southern California mainland, Northern and Southern Channel Islands, Baja California, and Mexico. So here's a map of the Channel Islands. The ancient individuals in the study are from the 6 main Channel Islands. The Northern Channel Islands are distinct from the Southern Channel Islands because inhabitants of the Northern Channel Islands, these 3 up here, when contacted by Europeans, were found to speak Chumash languages, which still exist today. And the Southern Channel Islands spoke Uto-Aztecan languages, which also exist today, but they were very different, distinct language families. So now we'll get into the results. Here, this is a phylogenetic tree that we created with outgroup F3 statistics, showing genetic similarities between different groups. As you can see here, the groups generally are clustering by geographical region. I just color-coded them so you can see that. But there is an interesting cluster that, and also lack of geographic continuity in here in the center, where is basically an early group of ancestry that I'll show you later, all lack of Mexico-related ancestry that comes into the later California groups. There's also some differences here between where people are not necessarily clustering by their geographic region, which is due to migration into the region, which I'll show you in other ways soon. Here's another way of representing the data called an MDS plot, which also shows clustering by geography. So first I'll start with the mitochondrial results. The mitochondrial haplotype results are really striking. When we look at individuals over 3,500 years old, 30 of 36 have the A2 haplotype, and the only ones without the A2 haplotype are all from mainland Southern California. And in contrast, after 3,500 years ago, only 35 of the 91 have the A2 haplotype, and many other haplotypes are represented at this time, pointing to possible migration from the region from outside. And in Mexico, very few of the Mexico individuals have A2 haplotypes, and they have many other haplotypes being represented. The Y chromosome results were also quite interesting. All except one of the Y chromosomes were Q1B1A. In contrast, South Americans in that prior study that I was talking about, the oldest ones had, a lot of the individuals had a very different Y chromosome haplotype, Q1A2A. And later on, they started switching haplotypes, which indicates genetic change that happened, which I kind of talked to with relation to the Clovis-related ancestry. So we wanted to do more rigorous analyses to determine if any ancestry came into California from outside. So to do this, we compared the genetic profiles of the oldest Santa Rosa individuals to the younger Santa Rosa individuals in other California regions as well. So we use a statistic called the F4 statistic to assess whether the younger Santa Rosa individuals had evidence of mixture from other groups. The F4 statistic is basically a allele correlation statistic where if you have mixture from another group and a genetic variance from another group, it'll create these correlations in the genetic structure that you can assess with this statistic. So it was interesting that the only groups outside of California with evidence of having mixed into California are a few Northwest Mexico groups. And interestingly, these groups are all ones that are in regions that now speak Uto-Aztecan languages, and they're of very similar ancestry to present-day people in the region. For example, the Odom or the Pima who speak a Uto-Aztecan language. And here's a graphic of this. On the y-axis is the amount of Northwest Mexico-related ancestry. On the x-axis is the age of the individuals. And so as you can see, the ancestry keeps increasing for all regions except for Central California. So the Northwest Mexico ancestry starts slowly around 5,000 years ago, increasing, and then increases over time throughout California. It reaches the highest levels in the Southern Channel Islands, which, as I said before, were known to speak Uto-Aztecan languages when the first Europeans met them. And still in that area, they speak Uto-Aztecan languages like Tongva. In contrast, the places where Chumash languages still have this Northwest Mexico ancestry, but not as much as in the other areas. And in Central California, there's less of this ancestry, but I'll get into a bit more of Central California later. So we also wanted to tell if the gene flow was primarily unidirectional or bidirectional. So we wanted to see basically whether the ancestry is coming just from Mexico-related ancestry into California, or is it also coming into Mexico, from California into Mexico. So one way to do this is we had actually 3 Mexico individuals who were older than 2,000 years old, and we compared them to the younger individuals, and we saw that the younger Mexico individuals had no evidence of California ancestry relative to the older ones. So this provides evidence against migration from California or the Great Basin as well, because we also tested that. It provides evidence against migration from California to the Great Basin during this time period. So this provides some evidence against the Central California or Great Basin origin of U.S. Tekken, because, for example, if there were people, their languages originating from there, you would think there'd be a lot of migration coming there over time. Probably also it'd be earlier than 2,500 years ago, but also you expect there to continually be migration coming down, and you'd be able to see it genetically. And so this provides some evidence against that, the Central California or Great Basin origin of U.S. Tekken languages. We also note that some of the prior claims, such as William Merrow's made some strong claims about the Great Basin hypothesis due to smaller mitochondrial studies that suggested that there was no evidence for migration from Mexico into the American Southwest. But here we're providing evidence that, in fact, there is ancestry coming in from Mexico to the American Southwest. So as noted before, there's some linguistic and archaeological evidence for Mexico influence in California, such as the contracting stem point. Our results don't explicitly rule out any of the previously suggested hypotheses, because you can basically find ways around these results, but they do require that many of them be revised. For example, if you wanted to believe in the origin from the maize agriculture, you would have to now change your hypothesis a bit, because maize agriculture was only present in the American Southwest after 4,000 years ago, which is after the inferred date of our U.S. Tekken related migration into California. So given we have evidence of migration that's happening at least 5,000 years ago, that's an argument against agriculture being the only plausible candidate for spreading a large number of people into this area. There's also evidence of migration into Central California at least 5,000 years ago, which is in line with the prior hypothesis of old U.S. Tekken language in that region. And this is all before maize agriculture is being spread into the American Southwest. So if you hold this hypothesis, you have to reckon with this. It doesn't explicitly rule that out, because perhaps you could have a situation where there's early migration of hunter-gatherers followed by a later migration of farmers who brought the U.S. Tekken languages. It's just that you would have to propose that situation instead of just a single migration bringing in all the U.S. Tekken languages. Yeah, another caveat is that the Mexico-related ancestry in California doesn't mean that the ancestry might have come directly from Mexico. It perhaps might have come in from Arizona and just was Mexico-related. It could theoretically have come in from the Great Basin as well. It might have come from Mexico up to the Great Basin and then down to California, but we need more individuals from the Great Basin to determine the route of migration into California. So now we'll look at this purple line, which is Central California. As I was noting before, the earliest Central California individual had evidence of U.S. Tekken related ancestry, which is consistent with the fact many archaeological hypotheses of old U.S. Tekken in the San Joaquin Valley before 5,000 years ago that was then displaced by these Yokutsan speakers. So there's evidence, some evidence archaeologically that people might have existed there. U.S. Tekken speakers might have existed there a long time ago. And now we find genetic evidence as well that there's ancestry there very early on. So now I'll talk about some of the population changes that happened in Central California. As a reminder, here are where the ancient individuals from Central California in our study lived. Most of them are on the coast, but we also have some individuals from inland in Calaveras County. So to assess what happened in Central California, we did our standard strategy of comparing the oldest Central California individual relative to the younger ones. And when we compared the oldest Pacific Grove individual over here to the later Central California individuals, the oldest Pacific Grove individual had a very interesting genetic affinity to some of the Paracues groups down here in Baja, Mexico. This one here is probably mislabeled, but that was the geographic coordinates that we got for them. It might have been that it was from a museum there, but they actually were from the tip of Baja California. But either way, that this is showing that there is a strong genetic affinity for these early groups in Baja California with the oldest Pacific Grove individual, and this ancestry started to decrease over time, starting with the later Central California individuals. So the Paracues were thought to have an ancient ancestry and potentially even an ancient language, and some suggest that there might have been an ancient language family in Central California as well before the introduction of the Yushin or Ohlone language family about 4,000 years ago. So this genetic attraction we observe of the oldest Central California individual to the Paracues individuals that's not found in the younger Central California individuals would be consistent with the Yushin migration into the region. But one possible source population for this migration might have been the Calaveras County here, but we need more sampling from this area to further test this, because we find evidence that there's migration coming in from here to here, but there might have been other areas that it could have come from as well. One thing we also wanted to address in this study was whether or not there was a genetic connection between Polynesia and California. So some people had previously suggested that there were ancient connections between Polynesia and California that led to the use of tomos in the Chumash and Tongva, and some even suggest that there are some linguistic connections. However, we find no genetic evidence for Polynesian ancestry in any of the California individuals. It's possible that there was some cultural connection that didn't involve mating between the groups, but this would not be seen in the genetics, and we don't observe it in the genetics. So next I'll talk about population sizes of ancient California. I'll get to those questions that some people have been asking already, but I'll get to them after the presentation. There are several different genetic ways to assess population sizes, but two commonly used ways are to use runs of homozygosity and conditional heterozygosity. So runs of homozygosity are regions of the genome that form when population sizes are not as large, such that parents are more closely related to each other on a genetic level. So more ROH regions indicate smaller population sizes. And the opposite of this is conditional heterozygosity, where which is smaller when populations are smaller. So there are two kind of independent ways to assess population size. So one thing we observe when we're looking at runs of homozygosity is that in this case, the higher bars are indicating more ROH regions, which indicate smaller population size. And as you can see, the northern and southern Channel Islands have more ROH regions than the mainland, indicating that they have smaller population size than the mainland, which is kind of what you'd expect because they're small regions, small islands relative to the larger mainland. And the population does seem to increase over time by, this is kind of organized by time within each region. So there's decreasing ROH in each region over time. So there's an increasing population over time. And when we calculate the effective population sizes using a maximum likelihood estimates, here are some of the estimates based on the younger individuals in each region. I'm only taking individuals who are less than 1,500 years old in each of the areas. And you can see that the Southern Channel Islands have the smallest populations, then the Northern Channel Islands, then the mainland, and then in Southern California and in Central California, and Mexico has the largest population. So interestingly, actually I'll get to that later. But one interesting thing is that when we compare the population sizes of California to other regions, the Channel Islands were more similar to Guam, Saipan, Patagonia, and pre-agriculture Caribbean, which are islands in general islands, except Patagonia is not an island, but it had a quite small population size. Similarly, the California mainland was slightly lower than the size of the post-agricultural Caribbean and Bolivia. And Mexico was more similar to Peru's population size, where Mexico and Peru are had some quite large empires at this time and had larger population sizes. And the order of these estimates is qualitatively the same as for the conditional heterozygosity. So conditional heterozygosity, as I said before, are inverse to the ROH estimates since they're inversely correlated. And so the estimates show kind of the reverse pattern, and so that's consistent with the similar population size estimates. One interesting thing is that when we analyzed different Mexico groups more in detail, we found that the more southern groups had larger inferred population sizes than the more northern groups. We don't have a really good interpretation of this, but it could indicate better agricultural conditions more south, which would allow larger population sizes. But we'll look into this more to figure this out. Lastly, I'll talk about the relationship of California and Mexico to the earliest sequenced Native Americans. In a prior paper we had previously published, we found that individuals from California, Nevada, and most of the earliest South Americans had ancestry related to this Anzac 1 individual, the Clovis culture individual, that was mostly displaced in South America. However, we find that in this study that ancestry is preserved in California, actually up to the youngest individuals. On the other hand, in Mexico, the Mexican individuals have no evidence of this ancestry, instead have a more basal ancestry that might have been from a different migration. So that's a very interesting finding that's also been found by a bunch of other groups. So we need to get more into this by having more older individuals from Mexico to start figuring this out. So in conclusion, ancestry related to Northwest Mexico Uto-Aztecan speakers came into California at least 5,000 years ago and increased in proportions over time, reaching highest level in Southern Channel Islands where the Uto-Aztecan languages are spoken. This is important because it impacts current models of the origins of the Uto-Aztecan language family, and currently provides most evidence for the origin being in perhaps Southern Arizona or somewhere around that region that comes before maize agriculture. But there are other hypotheses that are still consistent with the evidence. We find that population migrations occurred into the Central California coast, consistent with the arrival of the Uxin languages to the region and with prior genetic connection to an ancient language family. We find no significant Polynesian ancestry in the ancient California individuals, and we also find that population sizes of the Channel Islands was smaller than the mainland and similar to Patagonia and pre-agricultural Caribbean islands, but increased over time. We lastly find that ancient Mexicans harbor distinct ancestry from that found in ancient California and South America. So I'd like to thank the Chumash, Tongva, Ohlone, Essel, and Mexican communities as a whole for supporting and assisting the work, and in particular, Manuel Armenta, Rex St. Onge, and the late Ella Rodriguez, all of whom helped substantially with this study. I would also like to thank the members of the Reich Lab, and in this study, particularly John Johnson, Doug, and Brian at UC Santa Barbara, Paul Langenwater at Biola, and Jose Luis Punzo and colleagues in Mexico. So I can take any questions now, given it's 9 o'clock. There's other studies I could also talk about, but just wanted to stop there. Yeah.
Alrighty, let me go back up to the top. And anyone who wants to ask a question, go ahead and get it in now if you haven't already.
I can take this first one. The thoughts on the DNA evidence. The oldest migrations and sites into the Americas are claiming evidence back to over 25,000 years ago for the first migrations. It's difficult because we don't yet have the oldest DNA we have is only a bit over 14,000 years old. In terms of migrations that are extremely early, it's possible that there were migrations that we don't have their skeletal material yet, that we can't radiocarbon date per se. So it's a bit difficult at this time to tell. It's possible that these people also came into the Americas and then died off, so there's no genetic evidence of them yet. But yeah, genetically at least it's hard to don't have any clear evidence for that. There is one study about a possible this is related to a question about the South Asian ancestry. There is one study that actually a few that talk about a different sort of ancestry that's present in the Amazonians. That was a paper that was published in Nature, actually by our lab, and another lab then published a similar paper. This is a paper kind of suggesting that there's ancestry related to Papuans that is in people like Seruri or Kuritiana. Currently, we have suspect that is an artifact, but unclear right now. But that's still a possible thing, that there was another group of people that left some ancestry in Brazil and in the Amazon.
One thing, Nathan, can you quit sharing your screen so we
Yeah,
can
sorry.
see you bigger? Wonderful. And Karen thinks this is all mind-blowing and that mitochondrial DNA is where the promise is.
Yeah, mitochondrial DNA is very interesting because you can all see, you can see a lot from the maternal line. You can see changes over time. The problem with mitochondrial DNA is you're only seeing one side. You're only seeing the maternal side, and also you can't make inferences of admixture directly in an individual. So an individual would just have one of the mitochondrial haplotypes, and it can't have like two at once. It's a first approximation, basically. But so the autosomes, at least you can see where someone might have 30% ancestry of certain thing and 30% of another thing, whereas mitochondrial wise, you only get one of them. So like if someone, so like a good example is someone might have European fathers, a European paternal line the entire way, and have Native American maternal line the entire way. And in the maternal line, you would just think that they're pure 100% Native American ancestry because you're only seeing the mitochondria that's Native American. And you would be missing that there might be mixed with European or something.
Tina'd like to know, do you have any thoughts on the arrival origins of the haplogroup B? How far down in South America does it go?
Oh, interesting. That's a good question. There is some haplogroup. I'm trying to think of what the mitochondria results in the earliest paper, earliest South America paper. I think there were some B2 haplotypes, or just B in general haplotypes throughout South America. There was one of the interesting things was that we found that there was a D4H3A haplotype in Brazil very early on, which is similar to the one in, I think that was in the Clovis culture individual. But the B2, I can't remember exactly where it's found, but I think it was found throughout South America. I don't remember it properly.
And maybe I'm not supposed to announce this in a direct message to me. Laura said there was a major discovery of 5,500-year-old corn in December of 2019 in Las Capas. I won't say where, but it might be in Arizona, where 4,000-year-old sophisticated agriculture was previously discovered. This may not be published or announced yet.
Oh, interesting. Yeah, if you can demonstrate that maize agriculture started coming up very early and you start getting very early dates, you could have a scenario where you just had very early maize agriculture bringing in people with maize agriculture bringing in these ancestry types. Like say 5,500 years ago in Arizona, I guess presuming that people came in even earlier than that, there would have to be people that come in to that region as well as into California. It's possible, yes. Yeah, it's possible that if you have very early dates for maize agriculture, it could be that was a primary migration bringing in people. It's just you'd have to be very sure about those dates.
All right, Michelle's got a chapter in a book here. Thank you for this sweeping and fascinating talk. I particularly appreciate the consideration of indigenous oral traditions, which contain a lot of ancient knowledge, as one line of evidence about the past. Can you talk a little bit about more methodology? How does your team determine ancestry? For example, North Coast ancestry in the Chincha and Chile cases in the Andes. Do you use the earliest known individuals as a kind of baseline for the region? If your sample size is small and the DNA is degraded, how do you deal with those inherent challenges to reconstruct relatedness? What are the markers, features, or genes that characterize different regions, or do you characterize genetic ancestry in some other way, statistically or otherwise? Can you walk us through some of those methodological considerations?
Yeah, there's a lot of different questions there. So let's see where I should start. First, with the DNA quality. So the DNA is degraded, especially when they're older, but there are ways to get around this. So basically, the DNA is usually degraded at the ends, and there's also different computational ways and experimental ways to get rid of the degradation. So you can get rid of those bases that are degraded, because they're degraded usually in particular ways. The cytosines go to thymines oftentimes. So you're able to get around the DNA degraded patterns. It's also not that difficult in the Americas because there's nothing over like 20,000 years old. Whereas say like Neanderthals or ancient Europeans from 40,000 years old, or ancient Africans from 50,000 years old or something, that is much more degraded. So that is one problem, but it's not that big of a problem. We're usually able to figure that out. In terms of the determining ancestry, that's a really good question. So it's basically the same thing. You can kind of imagine what you would do when you're trying to study a population like let's say happen to know. Oh, sorry about that. Let me look at this slide. Okay. Let's say the ancestry. Let's say in New York what happened, and you're say an alien coming in New York, and you sequence people from 1,000 years old or 1,000 years ago or something like that. So when you're saying that there's an ancestry there that might be European at that time, you can't say that that's the only ancestry there. Like you can say that that ancestry was there, but maybe you missed that there was some Native Americans there, or vice versa. Maybe you only found Native American ancestry in New York and not in and no Europeans, but there were Europeans nearby. Or you have an artifact where you're only taking from a particular grave site, and that grave site is only people who are European or Chinese or whatever. So at very least, you can only say that at least that ancestry is there. In terms of determining what is an ancestry, ancestry is basically determined now that we have samples throughout the world of most people groups. You can basically tell in an unsupervised way what people are. So if you blinded me to say everyone in New York's ancestry, and you gave me lots of New York samples, I can tell you what general ethnic background they are, just because you're comparing it to people who exist either now in those regions or in the past in those regions. So you kind of are building a profile at different time periods. So say if you have a lot of, and then this gets better and better over time. So in Europe, say you have a profile of, say, Latvia 10,000 years ago, and 20,000 years ago, you have a different profile, and 5,000 years ago, you have a different profile. And you can kind of map out the region of what's going on and where people are. So as you get more and more individuals, you start getting more and more insight into what does ancestry look like on a broad level in that region at that time period. So that's basically how you're determining ancestry. It's kind of a statistical determination of the genetic variation there relative to other groups in that region or across the world. So yeah, that's about that. Markers.
Altart is asking, so have you found no evidence of any Southern Asian ancestry in any of the archaeological North or South American individuals?
Yeah, basically not, except for that that thing I was mentioning about the like Amazonian people who might have this, they call it a population Y ancestry. That is, it's not really South Asian ancestry, it's more Papuan or Ongi related, that are that some people think exists in certain groups, like in Brazil. I personally don't think that those genetic findings are robust, even though it was our lab who first proposed them.
Gail is asking about Polynesian presence in South America. Wasn't there the chicken?
Oh yeah. If you talk about more recently, yes.
Potatoes, and that's more recent.
Oh, well, there's two things about there. There is more recent ancestry from Polynesians, but the Botocudo, are you talking about the Botocudo example?
I don't know.
There was a Botokudo example where basically it was just an artifact of the fact that they accidentally they took museum samples of Polynesians as opposed to of people who were actually from the region. There's a subsequent study that was just published very recently about other Botokudo individuals from that region, and they're all Brazilian, Amazonian type ancestry. They're not Polynesian ancestry. So that was just a mistake that there just were Polynesian skeletal material there. But the only Polynesian ancestry you see in the South America comes much later, like not before, I think 500 years ago. Yeah.
And David would like to know, do you regard oral tradition as an empirical equivalent to DNA analysis?
Yeah, so oral tradition, there's a bunch of reasons it's helpful. One, for a fully different way of looking at the world. Like, so in terms of if there's different ways of looking at the world that are not just scientific or empirical. So oral traditions, in a particular worldview that it might be useful in seeing your ancestry or yourself through that lens. So that's a way in which it might be, equivalent in a different sense than DNA analysis or the scientific method. From a scientific perspective, the oral traditions also can be helpful in that they provide some, oral history is just one form of evidence that might be show that something might have happened because there might have been like a recording of it. It might also be, changed over time to fit certain narratives, but from a scientific perspective, it can provide at least some hypotheses for something. But I think the main thing about oral tradition is also that you can look at the world in a completely different way that's not scientific, and that could be just a different way of looking at the world.
And Marilyn, actually in a message to me, says the geography of different areas discussed may have influenced contact among peoples in different areas, for example, Mexico.
Wait, can you repeat that question again?
The geography of different areas discussed may have influenced contact among peoples in different areas, for example, Mexico.
Oh, I fully agree. Yeah. So the always throughout these places you have geographic, barriers or other reasons. If there's a lake there, if there's a mountain there, there's going to be difficulty traveling across the mountain. So you can usually see that genetically, especially in places where you have lots of DNA, like in Europe. But there are clear geographic barriers that cause people to go certain routes, and that you can see genetically because people are going to go, migrate in a certain way based on the geography. The only place you really see that right now is in obvious ways in the Andes. Oh, well, Patagonia too, right? Patagonia, people are going around the coast as opposed to like across the mountains.
Yeah.
So there's a bunch of other ways that manifest.
So we have a good question from David Young, but let's take a few other questions first and then come back to David, because I would probably ask David to unmute himself and interact with you directly.
Oh yeah, that's a good question. That's a good question that David's asking. Yeah.
Mary Pohl says all present-day Maya show genetic ancestry from South and Central America. Does this finding include the Huastec?
The what?
Huastec. They were the ones that were along the Gulf Coast north of the Olmec area.
So wait, what was the question again? Is the
all present-day Maya show genetic ancestry from South and Central America. But does this finding include the Huastec?
Oh, that's a good question. Well, we had a separate paper. I wasn't on it. I had helped at the beginning, but didn't end up on this paper on Belize Mayan groups and Guatemala, and because the papers I was on were very early Belize individuals. So yeah, I'm trying to remember about what happened with that group there. I think the Huastec might have had ancestry related to the Chibchan people from the south that came up after agriculture. that were affecting that region. I can't remember, but there's a paper by I think it was Doug Kennett and Mark Lipson in Nature Communications on Mayan in 2021 or 2020 or something, 2021 or something like that. Yeah, you can look that up, but I can't remember exactly.
And is it possible, Rob Wright would like to know, to calculate how many generations over time there have been with genetics?
Yeah, so the way you calculate generation time with genetics is usually basically a recombination rates or mutation time or mutation time. So the recombination rates and the mutation time are, to first approximation, pretty predictable. So there's a certain rate at which mutation happens over time or the recombination happens over time. So if you assume a certain amount of number of years per generation, then you can look at the number of recombinations that happen and see how many generations that happen. That's some way we use to infer admixture, basically, at what time the mixtures happened, or even what time the populations were a certain time of certain size. So you can infer a large population at a certain time and a smaller population at a certain time. And you can infer, if you have enough data, you can infer kind of when people are a particular population size and kind of when different events happen, when the mixtures happen. So yeah, that's basically the approximation of how you do it.
And Patricia says the connection between Calaveras County and the Central Coast in California is prior to European arrival.
Yeah, much prior. Yeah. So it's anywhere between 5,000 and 1,500 years ago. But that's pretty diffuse, obviously. That's like 3,000 years of time period. And but yeah, definitely much before Europeans came in. Yeah.
And Marilyn hopes that you will continue with your work in California, in particular the Nisean people who unfortunately lived in the epicenter of the gold rush. They're still struggling to be recognized by the federal government.
Yeah, I think this work is really important for bringing in, because a lot of the tribes, it's difficult with the state versus federal recognition and the recognition in general. So it's hard to use science directly to help people's recognition, but when we can, we want to do our best to try to help with the different tribes to get recognition, so they can get the resources that they need in terms of like the IHS, Indian Health Service money, or any sorts of other reasons. But I think it's really important in all these studies where the tribes are able to get sovereignty over their what they're doing and how they're kind of managing their land. Yeah.
Let's see, Soaring Bear has made a comment that corn spread from the Balsas down by the Ecuadorian coast includes 4383 before present Rio Grande. I'll be covering some of the domestication of corn next month in my program about the trade routes, and a lot to do with the maritime Pacific trade. And corn actually went back and forth a couple of times as it was domesticated to what then ended up in Mexico. But do you have any more thoughts? Or is there any kind of DNA that connects the Ecuadorian Pacific coast with?
Oh yeah, that's a good question. There are other people studying that right now. I don't know exactly the latest on that. Yeah, I'll kind of leave that where that is. I think that it's hard. I don't really know, because you kind of need ancient individuals from throughout the regions to figure this out, and at different time periods. And maize is kind of spreading at different time periods in different regions. And so I think there are studies now directly addressing that, but I don't know the results of those.
Thank you, Nathan, for this most excellent presentation. And Nathan is currently based at New York University and the New York Genome Center. To subscribe to free monthly issues of the Aztlander, magazine of the Americas, contact your host Jim Reed at mayaman@bellsouth.net. Thank you all for watching, and be sure to subscribe to our Aztlander YouTube channel.